Hello again, micro students, and get ready for your whirlwind tour of microbial biochemistry. This will be a very brief overview of the types of molecules that we see in cells, so we can recognize them and how they're going to interact as we move into the more biochemical reactions that we will be seeing in lab. This image is just to demonstrate that while biological macromolecules are special in how they come together and interact in a living system, in and of themselves, those molecules are not anything special. So Stanley Miller did an experiment where he put water, and that is methane, hydrogen gas, and ammonia into a chamber to simulate the atmosphere as it was on early Earth, water that had salt in it to simulate the ocean, and applied heat and an electric spark to simulate lightning. And what happened is amino acids were generated. So even though by themselves none of these chemicals are alive, no chemical is alive, they can be, in the presence of enough energy, brought together to be a molecule that is found in living systems. These are some examples of the molecules we saw on the previous slide. Carbon dioxide is a single carbon, double bound to oxygen on either side of it. These dots that are surrounding the oxygen atoms represent the unshared electrons. So there are still unshared electrons around this oxygen and that oxygen, and we have double covalent bonds on either side of the carbon. If we had stick and ball building models, the carbon would be the central atom and the oxygen's on either side, and there are two bonds. Each solid line represents one covalent bond. One covalent bond represents a shared pair of electrons. So there's two pairs of electrons on this side of the carbon shared with that oxygen atom. There are two pairs of electrons shared with this oxygen atom on the other side of the carbon. Ammonia, one nitrogen and three hydrogens. Again, these two dots are the unshared electrons. That has a high pH, so when we see molecules that have a lot of NH3s or NH2s on them, they will be alkaline. They will have a high pH. And then oxygen gas, O2, is two oxygen atoms with a double bond, so two shared pairs of electrons, and each oxygen atom still has two pairs of unshared electrons around them. Another representation of a molecule, this is methane. Carbon can bond with up to 4 other atoms because of the electrons in its outer shell. Carbon can bind with 4 other things. Hydrogen can make only one bond, and that single solid line represents a single covalent bond. Each covalent bond is that shared pair of electrons. This slide shows us 3 different sugars. I've got some boxes around functional groups that indicate where these sugars differ, and where they differ means they're also going to have different reactivities. These are glucose, galactose, and fructose. They all have the formula C6H12O6. They are called structural isomers, so that mer means a unit, so iso means same. So they're similar units. They have the exact same chemical formula. So if you were given only that formula, you would not know for certain which of these 3 you had in your test tube. You would have to be given more information. When we see the structure, glucose has this carbon double bound to an oxygen at the end, and the only other atom is a hydrogen. That is called an aldehyde functional group. Galactose has that same end on it, but here at the 1, 2, 3, 4th carbon, galactose has an OH to the left and a hydrogen to the right, whereas glucose at its 4th carbon has a hydrogen to the left and an OH to the right. And these are different. These are covalent bonds, shared electrons, so we can't just spin it around to make it be the same. They are fixed in place. Fructose, its 3rd through 6th carbon look just like glucose. The difference is at their 1st carbon, fructose has an OH, all single bonds around that carbon, and the 2nd carbon is where that double bond is found. And if we have a carbon double bound to an oxygen somewhere in the middle of a molecule, that is called a ketone. So I remember the difference between an aldehyde and a ketone, because a ketone is somewhere like a keyhole in the middle of a molecule, and aldehyde's the other one. You just got to memorize that. But because they have different arrangements of their atoms, these 3 sugars have different chemical reactivities and different binding capabilities. We have D-glucose and L-glucose. These have the exact same C6H12O6, but here the OH is to the right on carbon number 5 and it's to the left on carbon number 5 in the L form. And again, we can't just spin that around. These are called enantiomers. Chirality means that they are mirror images of each other. And so we can't just flip it over and lie them on top. We also have the difference here. So your hands are mirror images. I can face them toward each other and lay them on top. But that's not the same as if we have a chiral enantiomer molecule. If I take my hands that are facing the same direction and then superimpose them, they are not the same. I have to twist them. And you can't just twist molecules around. You can't swap where the bonds are. So they are mirror images. There are also enantiomers of amino acids. There's D-alanine and L-alanine. So the D-alanine is found in bacterial cell walls, and the L-alanine is found in human cells. My video recording is covering up the very first functional group. I've mentioned that you're responsible for the functional groups marked with red dots. So the first one is, and it is the aldehyde, because we have the carbon double bound to an oxygen, and the only other atom is an H, so that is the end of the molecule, and that is found in carbohydrates. The amino, we saw the ammonia was NH3. The amino functional group is NH2, found in amino acids, so that's in proteins. This capital letter R that shows up on all of these is for the rest of the molecule. So whatever other atoms are here is irrelevant to the functional group. The functional group is attached to the rest of the molecule. And that functional group is what gives that region of the molecule its identity and its function, its reactivity. Carboxylic acid, it's also called a carboxyl, is this entire thing. There is a carbon double bound to an oxygen, but there's also another oxygen and a hydrogen. It's found in amino acids, so that means it's in proteins, but that carboxylic acid is also in fatty acids. We're definitely going to see examples of each of these in the molecules later in this chapter. The hydroxyl is an oxygen single bound to a hydrogen somewhere on the rest of the molecule. So wherever you see just the OH, that is a hydroxyl. So alcohols have OH, monosaccharides, which are simple sugars, some amino acids, nucleic acids. Here's that ketone that we saw with the fructose, because the carbon double bound to the oxygen is that keyhole somewhere in the middle. We have R groups on either side. Carbohydrates. The methyl group is a carbon with 3 hydrogens attached. So methylated compounds. They'll be in alcohols and other organic molecules. We will also see methyl groups when we look at genetics and the control of how DNA gets expressed. Phosphate, very important in terms of molecules reactivity, and it is also a very key functional group that is found in nucleic acids, and it's part of our phospholipid cell membranes, and we will see it as an energy molecule, ATP. And that is a phosphorus with 3 oxygens and 2 hydrogens. We will see the actual drawn out structure of that. And lastly, the sulfhydryl. A sulfhydryl looks an awful lot like a hydroxyl. A hydroxyl was an OH. A sulfhydryl is an SH, found in amino acids and proteins. If you look at a periodic table, sulfur is directly below oxygen, so it has similar binding capabilities. The way we are going to build our organic molecules is primarily through a process called dehydration synthesis. Now, it's called that because water is produced as a byproduct. We don't literally suck water out. We don't dehydrate it, but we remove water from between 2 molecules to build a bigger molecule. So glucose in this case is the monomer, the single unit. We're going to build a polymer, multiple units, made of repeating similar molecules called the monomers. You're not going to have to memorize the structure of glucose. I would provide that to you and tell you it's glucose. But if you see an OH on one, and we have an OH on the other, but if we take just the hydrogen from this hydroxyl group and the entire hydroxyl group here, we have 2 hydrogens and 1 oxygen. That is where the water comes from. And then this carbon will bind to that oxygen, and that is the bond between 2 sugars and is called a glycosidic bond, because glyco is a term that means sugar. So glycosidic bonds are only found between sugars. Now if we attached a 3rd, 4th, 5th, etc. sugar onto this, each time we add an additional sugar, we form a glycosidic bond. There are different types of monosaccharides, single unit sugars. We can have glyceraldehyde. It is called an aldose. The suffix OSE typically means it is a carbohydrate. And we have an aldose because it has an aldehyde on the end. A ketose has a ketone in the middle. Now, the last 3 are telling us how many carbons there are. We have a triose, 3 carbons. It is also an aldose because of the aldehyde. This is a pentose, 5 carbons. Also an aldehyde because, or an aldose, because it has an aldehyde group. Hexose, 6 sugars, and this is an aldose because it has an aldehyde group. We could have a hexose that is also a ketose. That would just mean it would have a ketone group in it. They're classified by the carbonyl group, the carbon double bonded to the oxygen, and the number of carbons in that backbone. Again, I wouldn't expect you to know this is glyceraldehyde, but if I showed you the structure, you could tell me it's a triose because it has 3 carbons, and it is an aldose because of the aldehyde. We've seen the linear structures of the monosaccharides. In solution, they tend to form the ring structures. So A and B are both glucoses. They are just shown in different formats. So disaccharides mean we have linked two monosaccharides by dehydration synthesis, and there is our glycosidic bond. We see that oxygen linking two sugars. That is our glycosidic bond. If we link two glucose molecules together, we make maltose, the sugar that's important in beer making, adding malt. Lactose has two different monosaccharides, glucose and galactose. They both have that same C6H12O6 formula, and they are linked at a different position. Right? The two glucoses to form maltose are linked between, if we number in this direction, that's carbon 1, 2, 3, 4, 5, and carbon 6 sticks out. So this is carbon 1, and the other glucose is 1, 2, 3, 4. So we've linked from the first carbon to the fourth carbon. Between galactose and glucose, we've also linked between the first carbon and the fourth, but because the hydroxyls and the hydrogens are in different orientations on galactose, the bond goes upwards instead of downwards. And if we link a glucose to fructose, which still had that same C6H12O6 formula, the fructose, when it forms a ring structure, has 5 sides, and 2 of its carbons stick out. So we have carbon 1, 2, 3, 4, 5 between the first carbon of glucose and the 5th carbon of sucrose. Just showing you that depending on which two sugars are involved, the disaccharide is also going to look very, very different. Polymers, very large molecules of carbohydrates. We have 3 examples here. All of them are made of linking glucose over and over again. So starch is how plants store glucose, and it can be in the form of amylose, where we have a linear linking of the glucose molecules. We can also have amylopectin, where we have the linear links, but then we have branches coming off. All of these are, in the linear portion, are linked together like these two glucoses, from the first carbon to the fourth. In the branches, they're linked from the first carbon of one glucose to the 6th carbon of the other one, and that's what causes the branching. In animals, we store our glucose as glycogen. It looks very similar to amylopectin, but we have much more extensive branching, and we don't have a single linear form. So each long line is linked from carbon 1 to carbon 4. Every time we have a branch, at that branch point is from carbon 1 to carbon 6. Cellulose, found in plant and algae cell walls, is glucose, but it's a form called beta glucose. And in that case, again, there is a different position of the hydroxyl, and in cellulose, each glucose molecule is upside down relative to its neighbor. And that's what allows it to form these very linear, very rigid structures. All of these are made of glucose. So that is our examples of carbohydrates. There are a lot more and varied types of carbohydrates, but those are good examples, and they're the ones that we're going to need most when we're looking at metabolism of our unknowns in lab. Fatty acids are part of the category of lipids, and lipids are not polymers. They don't have one sort of monomer subunit that is linked together over and over again. They have components that are linked together, but then those components don't repeat. So this example is a triglyceride. Tri because there are 3 fatty acids, and glyceride because we have a glycerol. And a glycerol is a 3-carbon carbohydrate. Each glycerol has 3 hydroxyl functional groups. Each fatty acid has its own carboxylic acid functional group. Part of that is an OH, and that is where we get the dehydration synthesis. Each fatty acid links to glycerol by dehydration synthesis, so we would release 3 water molecules, and we have linked 3 fatty acids to glycerol. Now this whole thing is a triglyceride. The dotted line here just means it depends on the organism and possibly even the cell within an organism how many carbons are in that fatty acid chain. A phospholipid has that same glycerol. It has 2 fatty acids that were joined by dehydration synthesis, but on the 3rd carbon, instead of having another triglyceride, another fatty acid pointing downward, it has a phosphate group pointing upward. So there's our phosphorus with 3 oxygens around it with single bonds, and 1 with a double bond, and an additional R group. Depends on the organism what that R group will be. The key things with a phospholipid is that because of the phosphate, the top portion of a phospholipid is polar, hydrophilic, water-loving. The bottom portion has the fatty acids. They are hydrophobic, water-hating, or nonpolar. This phospholipid is shown with 1 linear fatty acid and 1 that is bent. And I've circled here that this one is bent because of a carbon-to-carbon double bond, and when that happens, it is called unsaturated. So saturated fats have all single bonds between their carbons. This little zigzag, each time there is a bend, that's a carbon, 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 carbon. The unsaturated fat has at least 1 carbon-to-carbon double bond. That causes a bend because a double bond means we're having to cram the 2 atoms closer together so that they can share 2 pairs of electrons. When we do that, we don't have as much room for hydrogens. Saturated fats, every carbon is saturated. It's completely surrounded by hydrogen. In an unsaturated fat, there's fewer hydrogens, so it is not saturated by hydrogens. So our phospholipids in a membrane look like this. We've got the polar heads facing into the cell, we've got polar heads facing out of the cell, and the fatty acid non-polar tails attract to each other because polar things associate with polar things, non-polar things associate with non-polar things. So if we were to put them individually just in an aqueous solution, meaning a polar water-like solution, they tend to form these liposomes. So S-O-M-E means bag of, so it's a bag of fat. We could have a lipid bilayer sphere, or we could have a single layer sphere. Cholesterol on the left, and then we have hopine on the right. The cholesterol is found in the cell membranes in eukaryotes, and more specifically, only in animals. And the hopine is found in the cell membranes of prokaryotes. They both have the significant ring structures. They have these very hydrophobic non-polar regions, all carbons and hydrogens. With cholesterol, we see at the bottom there's exactly one tiny polar region. This hydroxyl is 2/3 of a water. That makes it polar in this tiny region. Cholesterol is in our cell membranes. So the OH, the hydroxyl unit, associates with the phosphate, and the rest of the structure is sticking into that interior with the fatty acid tails. We do need cholesterol in our cell membranes. It reinforces the strength of our cell membranes. We just can manufacture all of the cholesterol we need. We do not need to ingest it in our diets. So high cholesterol typically results from ingestion of cholesterol that then the body doesn't have anywhere to put it, and it builds up in our arteries. The hoppine doesn't have any polar regions, so it is only in the fatty acid tail portion of prokaryotic membranes. My big warning, you do not have to memorize the R groups of amino acids. This is only 6 out of the 20 that are used in proteins, but that blue area tells you the R group for these 6 different ones. And we have one that's just a methyl group. This one has a sulfhydryl group. This has a CH2 and a hydroxyl group. This one has a carboxylic acid group. This has an additional amino, and so does this one. What doesn't change between every amino acid is this part. Let's see if I can actually draw on here. Oh, look at that. Yes, I can. This portion remains the same on all of the amino acids. So that is the backbone of each amino acid. Only their R groups vary. So when we see in the next slides, when we build a protein, we are never going to have reactions between the side chains, the R groups. The reactions are going to be between the carboxyl of one amino acid and the amino of the next. So this happens to be alanine. We have 2 of them. Here is the carboxyl group of alanine, the first one, the amino group of the second alanine, and we're going to do dehydration synthesis. So we're going to take one of the hydrogens off of the amino group and the entire OH off the carboxylic acid group. We release the water, and now here's the carbonyl carbon is still here. We're connecting from this carbon, to this nitrogen. We release the water. So this is called a peptide bond, and it is only found between amino acids, just like a glycosidic bond is only found between carbohydrates. This cartoon is showing the primary structure of a protein. Proteins do go through dehydration synthesis between amino acids to build, but that is not the end. With carbohydrates, once they are linked, that is the final perfect end of their structure. Proteins first have to do their peptide bonds, and those are covalent, to build the chain of amino acids. Primary structure. The secondary structure is once we've got that amino acid chain, how do the amino acids that might be very near or even kind of far from each other interact? They do that by hydrogen bonds. Right? The hydrogen atom attached to one portion of the molecule has a slight positive charge, and the oxygen the oxygen atom on another region is slightly negatively charged, and those dotted lines represent hydrogen bonds. And so the 2 types of secondary structure are an alpha helix, That's what that sideways fish is, the Greek letter alpha. That forms a coil. Or we can have a beta pleated sheet that is more ribbon-like. And this is all one primary structure. So one primary structure can have both alpha helix and beta pleated sheet. Just depends on the identity of the amino acids. The third level of structure is called tertiary. So we've gotten through the secondary. They haven't shown it, but this big chain could have both alpha helix and beta sheet secondary structures. And now we're going to sort of stitch those together into the final 3D shape. And that can happen by ionic bonds between an amino and an oxygen. We could have hydrophobic interactions. These carbons surrounded by hydrogens are very hydrophobic, so two regions that are very hydrophobic would be attracted to each other. We can have hydrogen bonding, and we can have covalent sulfur links. So two different amino acids that have sulfhydryl groups. The sulfurs can have a covalent bond. Because we can have so many different types of bond forming, means we hold this structure together very, very strongly. It takes a lot to undo the tertiary structure. All proteins must get through the tertiary structure level. Some proteins go to the fourth level, quaternary. Two or more completed tertiary structures have the same types of bonds in the tertiary structures, but they interact. This represents hemoglobin. It has 4 separate tertiary structures that had to go all the way to that level, and then those 4 tertiary structures interact to form the stable final molecule of hemoglobin. So there are 2 alpha chains, there are 2 beta chains. Hemoglobin has a lot of alpha helix structure, but it does also have some flattened beta sheet structure. So here is a summary of protein structure. The primary structure is the amino acids in order, peptide bonds that are covalent. Secondary structure is localized folding, hydrogen bonds. Tertiary structure is that 3D pattern that had all those different types of bond, the hydrogen bonds, the covalent disulfide bonds, hydrophobic interactions, hydrogen bonds. And then the quaternary structure, if the protein requires it, is 2 or more tertiary structures interacting. There's 3 examples of R groups here. You should be able to identify the R groups. And there are also the others in figure 7.6 that had the red dots next to them. The slides without my face and narration are also available in your module, so if you need just to look at those without the distraction of my narration, you can definitely do that. And nucleic acids are the 4th major category of macromolecules. We will cover those in chapter 10. But we do spend a little time with them in lab. In a few weeks we're going to have an experiment with that. And I did want to show just the basic structure. On the top, this is from DNA. DNA stands for deoxyribonucleic acid. And that is because the sugar that is involved is deoxyribose. There it is by itself. The other components of a nucleic acid monomer, there's also some sort of base, meaning a substructure that has nitrogens in it. There's also a phosphate. RNA, in the bottom series of pictures, is ribonucleic acid. And the difference is there's ribose in RNA, there's deoxyribose in DNA. And there's the difference. We have a hydrogen at carbon 2 on the deoxyribose. We have an OH, a hydroxyl group on carbon 2 in ribose. So it's deoxyribose because there is 1 fewer oxygen atoms. The other difference is in the base. DNA has thymine, RNA has uracil. So RNA never has thymine. DNA never has uracil. And both have 3 other bases that are called cytosine, guanine, and adenine. We will definitely cover way more about DNA structure in Chapter 10. This is the limit of what's required in Chapter 7. Yes, we're in Chapter 7. So this is also the end of the material for Exam 1. So we'll cover Chapters 1, 3, 4, 5, 6, and 7. Yes, it's a lot. I will be posting a study guide that will provide you with vocabulary, concepts you need, and thought questions. I sent out an email that had a lot more detail as to what to expect on the first exam. So make sure you check that out when it's posted, and there will be a discussion posted also for people who have questions about anything on that study guide.